Hello, my name is Elyse Schriber. I am an outgoing NNSA SSGF fellow, and this is my talk, Method Development for Serial Femtosecond Crystallography at X-ray Free Electron Lasers. To start, I would like to give a quick introduction about myself and my background. I recently completed my PhD in materials science at the University of Connecticut, where I graduated in December 2003. My PhD comprised of a lot of collaborative efforts between the University of Connecticut and SLAC National Accelerator Laboratory and Berkeley Lab. Most of my research was comprised of doing work at both synchrotrons and X-ray free electron laser facilities. And during my practicum at Lawrence Livermore National Laboratory, I worked on small angular X-ray scattering of biological molecules. So all of this work around using X-ray techniques at large user facilities led to me taking a position at SLAC National Accelerator Laboratory at the LINAC Coherent Light Source as a project scientist, where I have continued to advance new X-ray techniques for characterizing materials and biological molecules. So to start, there are two major national security threats outside of just weapons development and proliferation. Climate change is one of the major components. In 2008, the National Intelligence Committee noted that global climate change will have wide-ranging implications for the U.S. national security interests over the next 20 years. And we are starting to see the effects of these things today with these heat waves that are very much so affecting various parts of the United States, and extreme weather that is harder to predict. And then, of course, disease. We all lived during the COVID-19 global pandemic and saw the effects that it had on global security, national security, and even now afterwards, during the economic and national security implications of the COVID-19 pandemic. One of the major takeaways that they emphasized was that during the next 5 years after the initial 2020 shutdown, the COVID-19 pandemic is likely to exacerbate a range of global economic, human security, governance, and geopolitical strains. These two major global issues are national security threats for the United States and abroad, and also human threats on a very large global scale. So it's imperative that we start working on solutions to solve both these problems, or to at least find ways to ameliorate or navigate them as they happen. So the main way that my research and the work that I have done through NNSA and my graduate PhD was to find solutions for these problems via materials discovery. So within the two-dimensional materials, nanomaterials community, there are a variety of different materials and different types of classes of materials that can be used to aid in solving and also ameliorating both of these threats, including energy harvesting and storage using 2D materials, catalysis, which can help us be able to make cleaner burning fuels and cleaner burning tools, and also with biomedicine. So developing new materials to deliver targeted drugs, ways of being able to deliver vaccines in a more efficient way, also ways to store vaccines and be able to maintain their fidelity as we ship them around the world. So within material science, there's a huge array of different ways that we can use materials discovery and use new materials to solve and aid in ameliorating both these threats. So the big question of at the beginning is, how do we discover functional materials? There was a whole array of different materials listed on this infographic, and the big question of the day that I had at the beginning when I started doing research was, how do we discover functional materials? How do we actually say we know this material will solve a specific problem? A lot of the key to this is that the atomic structure, or the repeat unit in this material and at the atomic level, is the kind of golden ticket to discovering the functional relationship that we can to discover the function of that material. And it also helps us identify like how we can continue to make a new material that has a specific function if we can correlate the atomic structure to that material function. So on the left, we have two different materials that have two different types of functionality. And on the right, we have a whole array of different materials that have hybrid materials, so organic and metal organic materials, with a metal lattice and an organic ligand. By doing ligand exchange, we can change the photoemission quality and the photoactive quality of the materials, but we don't understand exactly why that happens. So the next question is, how do we determine the atomic structure of material? So over 100 years ago, X-ray diffraction was discovered by the Braggs when they were able to recreate the Young double slit diffraction experiment using a crystal lattice. So in this case, we have an example of table salt, so sodium chloride, and the table salt structure itself, at least the repeat periodic array of the table salt structure acts as its own double diffraction slit when exposed to monochromatic X-ray light. So an X-ray's wavelength is almost equal to that of the spacing or between the atomic lattices called d-spacing. And through a process of constructive and destructive interference, as the crystal is rotated at different orientations through a monochromatic X-ray beam, we start to see the reflections show up. These reflections are essentially a fingerprint of that atom's position in the lattice, and they change and move as different lattices become in the Bragg condition, or we start to see, and this is all a result of this constructive and destructive interference process. This GIF here shows how X-ray diffraction works. We start to see that different spots show up at different intervals, and this is a result of the crystal being rotated through the lattice. Another way to describe this is rotational crystallography. And from all of this information that we collect on 2D camera images, we can then use Fourier transformations to extract the three-dimensional information and know exactly where each atom is positioned. And we can do this with materials and with biological molecules. X-ray crystallography is a revolutionary technique that has really changed the way that we do everything from biomedicine to material science. The standard method for structure determination by diffraction is simply single crystal X-ray diffraction. You have a large, high-quality single crystal that you rotate through a monochromatic X-ray beam, and what you get is the same thing that we saw in the GIF of the previous slide, where we start to see spots appear and disappear as the crystal's rotated through the X-ray beam. This takes several iterations to be able to collect a complete data set, but is relatively straightforward, and there are actually tools now that make it almost like a pipeline where you don't even have to know what's happening. And what you get out is either some kind of file that you can then analyze, or you have a complete structure entirely. The other option, if you have microcrystals or very small powders, is you can have microcrystals on some kind of substrate that are randomly oriented. So rather than rotating the crystal through the beam, the X-rays themselves and the detector are rotated so that you're able to collect every possible orientation of the crystal. And instead of getting spots, you actually get rings as we're sampling the crystal at all possible orientations. So rather than having the crystal be rotated and you start to see spots appear and disappear, every possible spot is then shows up in a ring-like formation. We can take a 1D average, radial average of this 2D image, and then we get a powder diffraction pattern. Now, one can determine the 3D structure of material from this, but you have to have an accurate a priori model to be able to solve the structure, which relies on accurately modeling the position of the intensities here and the intensities themselves. Both of these methods kind of highly rely on two different things: large, high-quality crystals or having some kind of a priori model. There tends to be a big or small problem with both of these methods, in the fact that a lot of crystal materials, new materials and proteins, don't actually crystallize to become large single crystals, and we also don't have a priori models of them. They are new materials, they are unknown proteins, and the problem with this is that we can't use these standard techniques that are known. So how do we solve that problem? How do we work with these suboptimal crystals? We have two choices. We can either attempt to grow large single crystals, which can take years and can be a serious bottleneck to the materials discovery process, or we can find a way to deliver more X-rays to the crystal at a faster rate and be able to utilize these suboptimal crystals. So most of my research was spent at high flux X-ray light source facilities. Synchrotrons, which involve an electron beam that is in some kind of storage ring, where the electrons are then wiggled through a series of magnets. As they are wiggled, they're going near the speed of light. As the electron beam is wiggled, they produce X-rays, and the X-rays can then be collimated, and they can be run through a monochromator, so you can have one wavelength, and they end up at a beam line where you can do crystallography and a variety of other techniques. Synchrotrons at the Stanford Synchrotron Radiation Center, you can get about 10 to the 12 photons per second. Next generation synchrotrons like NSLS2 and MAX4 in Sweden, NSLS2 is in New York, can produce very highly coherent high flux beams that are much more highly focused, which allows you to actually be able to do more exciting crystallography and be able to work with these suboptimal crystals. The other option, when you actually cannot use a continuous beam, so if your sample is highly sensitive to radiation or you're trying to look at very fast time points, you can use an X-ray free electron laser. So rather than having a storage ring of an electron beam, you have an electron beam that is produced at an electron gun way down, at least a kilometer away from your experiment. And the electron beam is then accelerated through a very long quadrupole magnet undulator, 30 meters long in the case of the line at coherent light source at SLAC. What happens when it goes through this undulator is you have incoherent emission of X-ray light and randomly positioned electrons in the beam. As it goes through the undulator, the electric field starts to impart more force onto the electrons and the X-rays, and you start to see coherent emission of X-rays and also electron micro bunches that are spaced evenly. And we call this process self-amplified spontaneous emission. And when this occurs, you can get very, very fast X-ray pulses on the order of 30 femtoseconds. And you get a lot of X-rays in that pulse. So rather than having 10 to the 12 photons per second, you have 10 to the 13 or 10 to the 14 photons per 30 femtoseconds. So you're able to deliver X-rays to a crystal much faster and much more X-rays that are tightly bunched. So the XFEL is exponentially higher in terms of photon flux, and that's really important when you're trying to work with these suboptimal crystals. So biologists realized that there was a great use case for an X-ray free electron laser when they developed the technique serial femtosecond crystallography. In this case, suboptimal microcrystals are not placed on some kind of target, they're actually dispersed in the colloidal suspension. And the dispersant can be aqueous. In the case of biological molecules, it always is, but it could also be an organic solvent if you're working with materials. And this was kind of where my PhD work was really developed. I wondered if we could actually do the same thing with biology, with my materials that the biologists were already doing. Could we apply serial crystallography to material science? We had a lot of different microcrystals and suboptimal crystals that we couldn't get structures of. And the question was, could we actually apply this method? So the way that SFX works is that you deliver this like colloidal suspension to the interaction point of the X-ray, and you do this via liquid jets. So the jet velocity can actually be controlled such that you are delivering crystals at the same rate at which the X-rays are delivered. And the X-ray pulses can be delivered anywhere from 30 hertz, so 30 pulses per second, to 27,000 pulses per second at the high repetition rate facilities. And the way that SFX works is it combines both single crystal diffraction and powder diffraction. Rather than having one single crystal rotated through the beam, you have randomly oriented microcrystals that are delivered to the interaction point, and that pulse takes one partial diffraction pattern. So you get one single shot partial diffraction pattern of that one randomly oriented crystal. So rather than and then you collect thousands and thousands of these shots of randomly oriented crystals, so you can sample every possible orientation, but you don't know the orientation. It's random and basically produced by this dynamic dispersant that we use, the liquid jet. But these are all microcrystals, and if you were to take a projection of all the thousands of images that you get in an XFEL SFX experiment, it would start to look very similar to that of powder diffraction. So thousands of single shot images are collected during an XFEL experiment for the same reason why they're done, why you repeat a measurement during single crystal diffraction, because you want to be able to improve the fidelity of the measurement you're taking. The one other exciting thing about serial crystallography is that the temporal resolution of the XFEL is on the order of femtoseconds. At a synchrotron, you were oftentimes controlled by the actual bunches of electrons that are in the storage ring, and that is kind of basically ends at 500 picoseconds. So your time resolution is much, much slower than an XFEL. An XFEL can deliver a pulse on the order of femtoseconds. So over here, we can see that molecular movement in biological molecules and the materials and conformational changes in biological molecules is controlled near the nanosecond to microsecond timescale. So you can measure those things at a synchrotron. But if you're looking at atomic motion, like a photoactive, active molecule that has some kind of bond oscillation, you really are kind of stuck at the picosecond and femtosecond time range. And in material science, what we can do is potentially study things like exciton dynamics and other ultra-fast dynamics in materials by looking at structural changes. And what this could help us do is understand how to better design materials to be able to control those dynamics based on structural knowledge. So from that perspective, time-resolved SFX is very well suited for material science. And the way that it works is you use some kind of perturbation. In this case, it would be a laser pump. And then you have an X-ray probe. And the X-ray probe is so fast that it can actually capture almost a freeze frame of that photoactive state. And then you can start to then collect all of that data, pump probe data, and then compare it to a ground state structure. And it can tell you something about that excited state, where those atoms might be positioned, and more information about the transient process that occurs. So when I first started really getting interested in doing small molecule SFX, I realized that there was an incredible use case outside of my own group. We had a consistent problem of only forming microcrystals, and I didn't think that it was kind of localized just to our group. This is an example of some of the materials that we studied, because we were starting to see that beyond just one class of materials, you would start to see these microcrystals be formed. Many types of materials, it turns out, can be synthesized as microcrystalline powders, but are resistant to large single crystal synthesis. And while synchrotrons can now be used to do things like serial crystallography, we still run into a problem where a synchrotron cannot quite produce the amount of flux needed to measure a crystal that's 5 to 10 microns in size. So there's a significant need for small molecule serial crystallography in the material science and chemistry fields. One of the most significant differences between biological SFX and small molecule serial crystallography is that proteins have thousands of atoms in a unit cell. As you can see in this small video, these are all side chains. These aren't even atoms. So you have thousands and thousands of atoms, which results in much more reflections per partial diffraction pattern. As you can see, this is a proteinase K single shot pattern, and there are a lot of spots. Harder to see, but they're all there. And what that allows us to be able to do is be able to index this by using the periodicity of the already present spots. And it also relies on knowing lattice parameters of the repeat unit. And with biological molecules, it's a lot easier to attain. This allows us to be able to extract the crystal orientation that is lost during an SFX experiment. In small molecule SFX, you maybe have 30 to 50 atoms per unit cell. And what ends up happening is you have far less atoms far less reflections per shots, and no obvious form of periodicity. So the standard methods for indexing these single shot images falls apart when you start to have this very sparse pattern. And what ends up happening is you really can't identify any sort of real periodicity, and the method essentially falls apart at that point. So what we developed using an already made algorithm that was used once previously in 2014 in development of new techniques is we found that you could determine the lattice parameters from the XFEL experiment. So as we said, thousands and thousands of these randomly oriented crystals are sampled, and we get these single shot images. If you take 36,000 of these single shot images and you project them onto a detector model, you start to see what looks almost like a 2D powder diffraction pattern. Now, one could try taking the radial average of that powder pattern, but if you can see here, there's a lot of pathologies that show up, and this is because a lot of microcrystals are suboptimal. And what makes SFX unique is that we can work with these suboptimal crystals that are not as high quality as you would get from a single crystal experiment. What's not great is that the diffraction pattern itself reflects that poor quality by showing spot pathologies. The spots might be smeared. You can see some actually very robust smearing in this pattern here. And it becomes very challenging to extract unit cell parameters from that. So what we did is we designed a way to utilize tools that we already have at our disposal by being able to use an algorithm called Spot Finder, which we already used to find the spots in one of these single shot images. And then we can identify the D-spacing or the actual positioning of that spot in real space using the detector model that we already have. And then we can bend it down to a certain because there's a lot more pixels on the detector than we need. And we can start to form what's known as a virtual powder pattern, where we simply are using the intensities and the positioning of these spots from Spot Finder. And what that allows us to have is a powder pattern that's almost entirely free of any kind of instrument broadening. So it's very useful for now being able to get our unit cell parameters at the XFEL experiment instead of having to come with them to the experiment. Once we have a very accurate unit cell, we then are able to utilize the algorithm developed in 2014 called CZTBX.small cell process. And the way that the small cell algorithm works is that it uses a maximum clique algorithm to find the accurate nearest neighbor of the spots that we have available. And from there, it can recreate the periodicity that's needed to find the crystal orientation. So that in combination with the unit cell parameters and the correct crystal orientation allows us to index each of the frames properly. So the small molecule SFX pipeline has been extremely successful for my graduate research group, and it has allowed us to explain optical divergence in 3 materials. We had no idea why 2 were photoactive and 1 wasn't, and it turns out that the inorganic sublattice geometry is very different than the one that's not photoactive, and you can actually see that here. We lose the argentophilic silver-silver bonding elements that give these two their photoactive properties. And that really it answered a question that we hadn't been able to answer for 5 years. As our methods have become more refined and our synthetic strategies have advanced, we've also been able to show that you can actually do ligand exchange that can change the 1D and 2D quantum confinement in bulk materials. So normally the inorganic sublattice acts as a 2D quantum confined as a 2D nanomaterial. So it's quantumly, the electron wave function is quantumly confined to this inorganic sublattice. And we found that by doing ligand exchange, we can actually form almost inorganic nanowires in these 3D materials. And now we have one-dimensional quantum confinement. So a lot of really interesting science is being developed and discovered by way of using small molecule SFX. We've also been able to correct literature values on inorganic sublattice organization in some silver alkane thiolate 3-dimensional structures. And this is partially because of the challenges in being able to, in using powder X-ray diffraction, where you have to have a correct a priori model, and it's not always easy to have one of those. So what small molecule SFX has allowed us to do, and what we believe the rest of the community should be able to access, is a way to do rational materials design. And if we're going to solve these major problems facing our world, for instance, climate change and disease, we really have to be able to take a rational approach to materials discovery, as opposed to waiting, years to be able to identify a way to be able to characterize a new material. Now we can all of a sudden say very clearly, okay, if I want to design a new material with these specific kinds of properties, I now have a structural database that I can use, and I can figure out exactly what kind of structure I would need to attain that particular property. And this is what leads the structure-function relationship of rational materials design. One of the major challenges in small molecule SFX that we've found over the years is that sample delivery, this dynamic sample delivery with liquid jets, is extremely difficult. So the way that sample delivery works is you have a helium focusing gas and a liquid capillary, and the sample is delivered via the liquid capillary. The helium focusing gas is then delivered as well. And at the exit aperture, the gas acts as a focusing element to the jet. So as it comes out of the aperture, you get a nice conical jet by way of the helium focusing gas. And what this allows us to do is reduce our sample consumption and also reduce the amount of background scattering that you get from the solvent itself, which for biological science molecules is very important. For material science, it's not as challenging as we tend to have higher scattering factors. One of the difficulties of liquid jets is that we run into a significant problem with jet instability. The XFEL beam, which is about 1 to 3 microns in size, has to overlap with the jet, which is usually about 5 to 10 microns. And we can't see the X-ray beam, and we can barely see the jet using optical cameras inside the hutch. So a lot of times a person has to be consistently modifying and moving this jet to be able to find it, position it correctly as it changes, because it is a constantly changing system. And it moves on the scale of microseconds, which our eyes can't even keep up with that. So we're always kind of a little bit behind the experiment. And I heard a great quote from a paper that I was reading, that there should not ever be any thinking done at an XFEL experiment because we can't think fast enough. And that's true. An XFEL is so much faster than us, and every single problem that arises, we're seeing it far too late. The other problem we run into is that some crystals, especially needle-shaped crystals, tend to actually preferentially orient as a result of the forces that the jet itself is placing upon them. And what that means is that we have to then sample more and more of that particular crystal because we can only get we get only a specific orientation. Generally, the crystal hit rate, so how many actual diffraction patterns we're getting that we track via spot finder, is between 0 and 20% on average throughout an experiment. And what this has led to is a lack of high throughput data collection. And if we're going to turn this into a useful method for rational materials design and that can keep up with the rate at which materials are being synthesized, we need to create a high throughput method. So to solve that problem, we started looking elsewhere beyond the liquid jet sample delivery, which is the most common way to deliver samples to an XFEL. And we looked to Cyanion, which is a company that actually makes droplet on demand systems for inkjet printing. And this particular droplet on demand system has been modified to actually be useful for XFEL data collection. And it was initially purchased with the idea of making it easier to do XFEL experiments in a wide variety of biological sciences research. But we thought maybe this would become a useful technique for small molecule crystallography as well, because we have so many samples that need to be measured, and we don't have a high throughput method for data collection or sample delivery. What makes the Cyanion droplet on demand so unique is that we have a 96-well plate that can be placed inside a cooling plate, and we have a robot that can actually move the nozzle, which is controlled via piezo actuator. It can pull up sample at a specific volume, and then using this piezo actuator system, it can emit droplets that are perfect, that are the exact same shape and the exact same volume. And it can be synchronized perfectly to the X-ray pulse delivery. So anywhere from 120 hertz to 10 kilohertz. We tested this method for small molecule crystallography using a previously characterized sample, and we found that the hit rate went up to 30% in one run. Usually it takes us several go-arounds with liquid jet sample delivery to attain that. So to immediately be able to get a 30% hit rate was really exciting for us. It made us think that it was possible to actually do high throughput data collection with SFX, with small molecule SFX. And this is just an example of the droplet itself, and then the explosion from the XFEL pulse. So we can synchronize each pulse to the droplet, which is also exciting because it means we're using less sample and we're also producing less waste. So as a result, this led to the first service small molecule SFX experiment at the LINET Coherent Light Source. Synchrotrons all over the world offer service crystallography, but XFELs normally don't. There's a much higher barrier to entry at an XFEL. Therefore, being able to offer a service to people really lowers the barrier to entry and makes the XFELs high throughput measurements available to more than just experienced user groups. So in this case, we have a very small glass nozzle that pulls up sample from the 96-well plate. It then goes and delivers that sample to the interaction point here. And then we were able to collect data for a certain amount of however long we need to. We can screen for 3 minutes. If the sample is not diffracting well, we move on to the next sample. What makes it also exciting is that we can actually there's a washing station with a Cyanion, so we can actually work between samples and solvents rapidly without worrying about sample contamination. So as a result, from this very first service as small molecule SFX experiment, we were able to screen 86 samples of a variety of different types of materials, from hybrid materials to organic materials to inorganic materials, using the 120 hertz, so 120 pulses per second LCOS. It allowed us to solve structures in a very fast time as well. So 15 minutes of diffraction data was only needed to solve one structure, and we were able to, and in 66 minutes straight of diffraction data collection, we were able to solve a structure. This is much faster than our previous rate, where we oftentimes are stuck in a 3-hour waiting period dealing with sample delivery challenges. We still have several data sets we're still working up, and it is still a work in progress. But this experiment allowed us to collaborate with 25 user groups in 5 different countries in only 60 hours of beam time. And this is just a list of the user groups we were able to work with. So this result really showed us that by developing a technique, which was my PhD work, and then opening it up to a larger population of users, we actually were able to offer people an opportunity they would normally never get access to. So the reason that this work is so important in regards to this big overarching problem of solving these two national security threats of disease and climate change is that when we start offering a service at a facility like an XFEL that can deliver X-rays so quickly, it can produce data so fast, is that structured solutions are the first step in materials discovery. So if we can do small molecule SFX and get people's structures into 15 minutes, we are now completing the first step in materials discovery in a much faster rate. It took us 5 years to be able to solve the structure of one of our materials. And if it hadn't been for the development of small molecule SFX, that would have never happened. And now we're able to offer people this method and get them a structure in 15 minutes. It has the potential to also be a revolutionary technique, just as a service technique for both material science and chemistry. So if we think about small molecules used in drugs, if you have a powder as opposed to a single crystal, rather than wrestling with NMR or having to produce large bulk amounts of a novel small molecule, you can send a small amount to an XFEL and be able to get that structure in a much faster way. The hope is that a service small molecule SFX tool will scale up the rate at which we discover new materials and the rate at which structures are published. And what we hope that will do is then help us create a high throughput pipeline that will be able to utilize the next generation of XFEL facilities like LCLS2 that have a high repetition rate. As I mentioned earlier, we can go from either 30 shots per second or 27,000 shots per second. I know that normally in things like hydrodynamics experiments at some of the other defense labs, that 7 or 8 shots or 41 shots per year is huge. But in the world of XFELs, 27,000 shots per second is kind of where our limits are reached. But we really need to develop methods and sample delivery methods that can actually keep up with the rates at which we can deliver X-rays. And I think that service crystallography at XFELs, especially for the material science and chemistry communities, is an answer to that question. And I'd like to acknowledge that all of this research was made, all of my graduate research and the work that I've done collaborating with various groups is a huge, it was a huge group effort by a variety of different people at UConn, at LCLS, at Berkeley Lab. We even had graduate students from Stanford and Berkeley that were also helping with this. And the NNSA SSGF also funded my graduate research and allowed this work to be possible. If anyone is watching this talk and has a collaborator or they themselves are synthesizing new materials that need structures and you are not getting it anywhere else, you're having trouble, please look me up on the NNSA SSGF Outgoing Fellows website and email me, because we're actively looking for users that are interested in utilizing this technique. Thank you so much.